Today, we're going to delve into the fascinating world of the nervous system, focusing specifically on the spinal cord and its associated spinal nerves. Our material for this discussion comes from Professor Vanessa's lecture presentation, which provides an excellent foundation for understanding these critical components of our biology. We'll start by looking at the basic structural and organizational characteristics of the nervous system as a whole. Then we'll discuss the structure and functions of the spinal cord itself, including the three protective meningeal layers that surround it. We'll explore the distinct roles of white matter and gray matter in processing and relaying sensory information and motor commands. We'll also break down the major components of a spinal nerve, understand what a nerve plexus is, and connect the distribution pattern of spinal nerves to the body regions they innervate. Moving forward, we'll examine the significance of neuronal pools and the major patterns of interaction among neurons within these pools. We'll also describe the steps involved in a neural reflex and classify the different types of reflexes. Finally, we'll distinguish among the various motor responses produced by reflexes and explain how reflexes interact to create complex behaviors, ultimately touching upon how higher brain centers control and modify these reflex responses. Let's begin with the big picture, the structural organization of the nervous system. We can broadly divide it into two main parts. First, there's the central nervous system, or CNS. This includes the brain and the spinal cord, and it's essentially our body's processing center. It's where all the decisions are made and information is integrated. Then, we have the peripheral nervous system, or PNS. This system consists of the cranial nerves and the spinal nerves. It's responsible for the sensory input that comes into the CNS and the motor response pathways that carry commands out to the body. A key concept when thinking about the nervous system is reflexes. These are quick, automatic responses triggered by specific stimuli. An important type are spinal reflexes, which are controlled solely by the spinal cord without direct input from the brain. They're incredibly fast because the signal doesn't have to travel all the way up to the brain for processing. Now, let's focus on the spinal cord itself. This vital organ is made up of nervous tissue, and it's carefully housed within protective membranes, which we call meninges, and, of course, the vertebral column. Its main job is to carry sensory and motor information between the brain and most other parts of the body. It's also the origin point for many of our spinal nerves. Looking at the gross anatomy of the spinal cord, it's typically about 45 centimeters, or roughly 18 inches in an adult. It's not uniformly wide. At its maximum, it's about 14 millimeters, or half an inch. It extends from the brain down to the first or second lumbar vertebrae. Inside, it contains both gray matter, which is where you find neuronal cell bodies, and white matter, made up of myelinated axons. The spinal cord is divided into four main regions: cervical, thoracic, lumbar, and sacral. It has 31 segments, with each segment giving rise to a pair of spinal nerves. The cord also exhibits bilateral symmetry, meaning it's mirror images on either side. You'll notice grooves that divide the spinal cord into left and right halves. On the posterior side, there's the posterior median sulcus, and on the anterior side, a deeper indentation called the anterior median fissure. Running through the center of the spinal cord is the central canal, an internal passageway that contains cerebrospinal fluid, or CSF. The spinal cord isn't a uniform cylinder. It has areas called enlargements, which are visibly wider. These regions supply the limbs and therefore contain more gray matter. For instance, the cervical enlargement supplies nerves to the shoulder and upper limb, while the lumbosacral enlargement supplies nerves to the pelvis and lower limb. At its distal end, the spinal cord tapers into a conical shape known as the conus medullaris, located below the lumbar enlargement. Extending from the conus medullaris, you'll find a bundle of nerve roots called the cauda equina, which means horse's tail in Latin, due to its appearance. Finally, there's the filum terminale, a thin thread of fibrous tissue at the very end of the conus medullaris, which attaches to the coccygeal ligament anchoring the spinal cord. Spinal nerves are formed by two roots that attach to the spinal cord. The anterior, or ventral root, contains the axons of motor neurons, carrying commands away from the CNS. The posterior, or dorsal root, contains the axons of sensory neurons, bringing information into the CNS. These spinal nerve roots actually divide into smaller rootlets before entering or leaving the spinal cord. You'll also find spinal ganglia, also called dorsal root ganglia, which house the cell bodies of these sensory neurons. They're situated between the pedicles of adjacent vertebrae. Spinal nerves are formed by the union of these posterior and anterior roots. They emerge as pairs from each side of the vertebral column. Crucially, all spinal nerves are mixed nerves, meaning they contain both afferent or sensory fibers and efferent or motor fibers. These nerves then branch out. The white ramus communicans contains myelinated axons that innervate glands and smooth muscle. The gray ramus communicans contains unmyelinated fibers that also innervate glands and smooth muscle. The posterior ramus innervates the skin and muscles of the back, while the anterior ramus innervates the ventrolateral structures of the trunk and the limbs. Naming spinal nerves is fairly straightforward. They're designated by their vertebral region and number. There's a slight trick for the cervical nerves. C1 runs above the first cervical vertebra, but C8, our eighth cervical nerve, is actually below the seventh cervical vertebra. All other spinal nerves are named for the vertebra directly above them. So, for example, T1 is found below the first thoracic vertebra. The spinal cord is protected by specialized membranes called spinal meninges. These provide physical stability, absorb shock, and create a pathway for blood vessels that supply the spinal cord. These meninges are continuous with the cranial meninges that surround the brain. There are three layers, from outer to inner: the dura mater, the arachnoid mater, and the pia mater. An infection of these meninges, whether viral or bacterial, is known as meningitis. The dura mater is the tough, outer fibrous meninx. It contains dense collagen fibers oriented along the longitudinal axis of the spinal cord. It's continuous with the cranial dura mater, fusing with the periosteum of the occipital bone, and it also fuses with the connective tissue around spinal nerves. Its distal end tapers into a dense cord of collagen fibers, blending with the filum terminale to form the coccygeal ligament. Between the vertebrae and the dura mater, you'll find the epidural space. This space is superficial to the dura mater and contains loose connective tissue and adipose tissue. This is a clinically important area, as epidural anesthesia is injected here to provide regional pain relief. Deep to the dura mater is the subdural space, which is typically a potential space, meaning it's usually collapsed unless there's fluid accumulation. Next is the arachnoid mater, the middle meningeal layer. It's in contact with the inner surface of the dura mater. This layer includes the arachnoid membrane, which is a delicate web-like layer of simple squamous epithelia, and the arachnoid cubeculi, a network of collagen and elastic fibers that extend between the arachnoid membrane and the innermost pia mater. The space between the arachnoid mater and the pia mater is called the subarachnoid space, and it's filled with the arachnoid trabeculae. This space contains cerebrospinal fluid, or CSF, which carries dissolved gases, nutrients, and waste products. A lumbar puncture, often called a spinal tap, involves withdrawing CSF from this subarachnoid space. Similarly, spinal anesthesia involves introducing local anesthetics directly into this space. Finally, we have the pia mater, the innermost meningeal layer. This is a mesh of collagen and elastic fibers that is firmly attached to the underlying neural tissue of the spinal cord itself. The blood vessels that supply the spinal cord actually run along the surface of the pia mater. Paired denticulate ligaments, which are extensions of the pia mater, anchor it to the arachnoid mater and the dura mater, helping to prevent any lateral movement of the spinal cord. Now let's explore the rules of gray matter and white matter. Gray matter is composed of neuronal cell bodies, neuroglia, and unmyelinated axons. It's where information is integrated and commands are initiated. In the spinal cord, gray matter is located deep within. White matter, on the other hand, consists of both myelinated and unmyelinated axons. Its primary function is to carry information from one place to another. In the spinal cord, white matter is superficial. The organization of gray matter is quite specific. Masses of gray matter within the CNS are called nuclei, and in the spinal cord, they're organized into regions called horns. We have posterior horns, which contain somatic and visceral sensory nuclei, receiving incoming information from receptors. The anterior horns contain somatic motor nuclei, sending outgoing information to our effectors. And in the thoracic and lumbar segments, you'll find lateral horns, which contain visceral motor nuclei. The exact location of a sensory or motor nucleus within the gray matter determines which body part it controls. Connecting the two sides of the gray matter is the gray commissure, a narrow band of gray matter surrounding the central canal. White matter is also organized into specific regions, which we call columns. There are three main columns: the posterior white columns, located between the posterior horns and the posterior median sulcus; the anterior white columns, found between the anterior horns and the anterior median fissure. These anterior white columns are interconnected by the anterior white commissure, where axons cross from one side of the spinal cord to the other. Lastly, the lateral white columns are situated on each side of the spinal cord between the anterior and posterior columns. Within this white matter, we find bundles of axons called tracts. These tracts are crucial because they relay the same type of information in the same direction. Ascending tracts carry sensory information up towards the brain, while descending tracts carry motor commands down from the brain to the spinal cord. Let's move on to spinal nerves and plexuses. Spinal nerves are surrounded by three connective tissue layers. The outermost layer is the epineurium, a network of collagen fibers. The middle layer is the perineurium, which separates the nerve into bundles of axons called fascicles. And the innermost layer is the endoneurium, which surrounds individual axons. A pair of spinal nerves emerges laterally from each spinal cord segment. They form from the junction of the anterior and posterior roots. And as we've discussed, all spinal nerves are mixed nerves, containing both sensory and motor fibers. Peripheral nerves, on the other hand, are formed as spinal nerves branch and interconnect. These are also all mixed nerves, and they share the same connective tissue layers as spinal nerves, as these layers are continuous with one another. Understanding the peripheral distribution of spinal nerves introduces us to the concept of a dermatome. This is a specific bilateral region of skin that is monitored by a single pair of spinal nerves. Dermatomes are clinically very important because damage or infection of a specific spinal nerve or spinal ganglion can lead to a loss of sensation in its corresponding dermatome. For example, peripheral neuropathies are regional losses of sensory and motor function, often resulting from trauma or compression. A well-known example of this is shingles, where the varicella zoster virus attacks spinal nerves and produces a painful rash along the corresponding dermatomes. In summary, we've explored the intricate structure and vital functions of the spinal cord and its associated nerves, from the protective meninges and the distinct roles of gray and white matter to the detailed anatomy and clinical significance of spinal nerves and dermatomes. This understanding is foundational to appreciating how our nervous system processes information and commands our body's movements, as outlined in Professor Vanessa's comprehensive lecture. If this discussion deepened your understanding of the nervous system, share it with someone who also wants to learn more about the spinal cord.